Well, welcome to everybody who is joining the seminar. We are very sorry for these technical troubles. What we have learned that if you would like to use Zoom and be sure that you will be visible, you will be able to share your presentation. Don't use Ubuntu or something like this. So it is a lesson for the future from these technical troubles we had. In order not to waste our time, I would like to introduce the speaker. The speaker is Cemal Koba, who at that moment is a postdoc at the computer vision team at Sano. And Cemal has completed his PhD study in computational neuroscience in IMT Lucca in Italy. He is mainly interested in the methods of processing and detailed analysis of neuroimaging data. And what he will present today will be one of the results of his research in Sano. So, Cemal, floor is yours. Screen is yours.
Thank you, Marian. And yes, sorry everyone for the delay. So today I will talk about some of the research outputs of mine during my time in Sano. We worked on an MRI, fMRI data set of stroke, and we generally investigated the effect of stroke on functional brain connectivity. To make a quick description, stroke occurs when blood supply to arteries are blocked. Generally, it is due to the lack of oxygen. And due to this lack of oxygen, brain tissue can be damaged, and it can die in a very short amount of time. And of course, this lack of blood supply will cause tissue deformation and functional and behavioral impairments related to the region of this stroke lesion. And it presents a good model for brain functionality because depending on the stroke location of the stroke lesion, you can make predictions or you can investigate the brain function. And if you can record the recovery progress, if you have longitudinal designs, you can also keep track of recovery and behavioral improvement across time. So it is also conveniently easily detectable from MRI, magnetic resonance imaging. On the right side, you can see different contrasts for magnetic resonance imaging, and in almost each of them, stroke lesion is identifiable by naked eye. Also, you can compare the brain function with stroke patients compared to healthy controls using functional magnetic resonance imaging, which is useful for recording time series from brain. However, first, when we check the stroke patients with MRI, we can see that although stroke at first sounds random stroke lesion. Actually, it is, let's say, pseudo-random. And stroke lesions are concentrated mostly in subcortical areas around basal ganglia and thalamus. So although some subjects will get away with damage around here, in the bigger picture, when we investigate the effect of stroke, we are mainly investigating the effect of subcortical ischemic damage on the brain function. However, all the subjects have different effects, so it is it's hard to make group level analysis in this scenario. And in the pictures, you can see the mean stroke lesion overlap from two different samples, one with relatively slow, but one with quite high sample size. They are mostly gathered around the same region that correspond to middle cerebral artery. And you can also record the brain signal, bolt signal from stroke lesion, and it's been used very commonly in the previous MRI research to understand the brain function after the stroke lesion. However, you cannot expect to have the same kind of signal after the stroke lesion, because as you can see on this image, on the left side, there is the bolt signal, a percent change in the bolt signal from control subjects. And on the right side, you can see the bolt signal from stroke patients. And you can easily see that the signal is more heterogeneous, more variance. There is more variance, but there is still some kind of pattern. It shows that the signal is still there, but it is distracted. In theory, though, since there is no oxygen supply and bold signal actually is essentially measuring the oxygen consumption, there should be zero activation from the stroke lesion. But there is some signal, and there looks to be a pattern, probably corresponding to the task in this study. So it is either quite noisy or needs some improvement to be compared to the bolt signal of controls. And on the right side, you can see, although it is not directly from lesioned area, since there is some disconnection between two regions, bolt signal is still affected by the distant lesion. So it doesn't have to be the signal from the lesion, but there are distant effects, long distance effects of stroke lesion in brain function. Another common finding from stroke research is that intrahemispheric connectivity decreases, and so interhemispheric connectivity decreases and intrahemispheric connectivity increases, meaning that the communication between hemispheres decreases and hemispheres become more autonomous. On this study, for example, you can see lesioned hemisphere is giving less signal to a certain task. However, healthy hemisphere is trying to compensate for the lack of neuronal response. So considering this heterogeneity challenge, noisy signal and intrahemispheric puzzle, in this study, we aim to first make stroke data, functional stroke data, comparable to that of control cases. Because generally, the common approach is to mask the signal around the stroke lesion to avoid this noisy signal. However, no one wants to lose the data that's already there that can be salvaged. Also, the disruption is not unique to the stroke lesion, and there are like long distance effects that is due to the stroke damage. In order to do whole brain comparisons and to identify the real, not real, but better amplified signal that reflects the neural plasticity due to stroke, like how brain reorganizes itself as a response to stroke damage. Whole brain comparisons are needed, but to do that first, these noisy data has to be improved. So first we wanted to make sure that this data is salvageable and can be used. And then we wanted to check the lateralization of the brain under these new resolved, improved signal conditions. And we wanted to check how these functional connectivity deficits are related to behavioral impairments. And we used a dataset from Washington University. And in the dataset, the distribution of the stroke lesion is quite similar to the other studies. So it reflects the average distribution that's around basal ganglia. And we processed this data according to traditional resting state pre-processing pipeline. And we prepared the data for further analysis. I don't want to give too much details for the pre-processing here, but also the stroke data are pre-processed as if they are data from control. And after this step, we tried to investigate the noisiness of the stroke data so that they are like more in line with the control set. And to do that, to fix the bolt signal, we used a trick that is already defined in the literature. Bolt signal from fMRI data has this temporal confound that shows the blood flow from internal carotid arteries to venous sinuses. And this is actually detected relatively easy. You need to make a cross correlation design with a reference signal. So first you need to define what is the reference signal. It can be mean signal from internal carotid arteries. It can be the mean signal from whole brain, or it can be the mean signal from venous sinuses, just to have a temporal reference point. For example, in the image, you can see a study using a reference signal from a whole brain, basically the mean time series from the whole brain. And for each voxel, they just temporally move the signal, plus minus maybe, I don't know, 6 to 8 seconds, to see what is the time shift that satisfies the highest correlation with the global signal, the reference signal. And the time shift time point that gives you the maximum correlation actually reflects it's very similar to the time lag from other modalities such as perfusion and dynamic susceptibility mapping, although it's a bit more noisy. And this delay mapping can be used to track the information flow. So you can see there is a flow from again internal carotid arteries to venous sinuses that reflects the susceptibility mapping, and there are also other studies showing with the perfusion. So this can be used as a time reference for the signal. And shifting the time series to eliminate the delay actually improves the data quality and improves the, for example, explained variance in the models that they are using. For example, the study that is the figure from on the right side, used seed-based functional connectivity for posterior cingulate cortex, and they showed that the connectivity map has higher explained variance when they account for this temporal delay. So in general, in fMRI studies, it is better to pay attention to these information flows, and when you fix the temporal differences among between slices, you will have a better explanatory power for your model. And when you do this for stroke patients, the most extreme time points will point to the stroke lesion. This is probably due to the fact that stroke lesion has lower or delayed blood flow thanks to peripheral blood support or weak blood support. So the blood will arrive there later. Therefore, the most delayed voxels will be related to lesion, right? And this has been also described before, but this method has not been used extensively for whole analysis. So this is mostly used to actually define the lesion area from fMRI signal. But since it improves the data for healthy controls, we also wanted to use this model to improve the data of stroke subjects as well, and wanted to move forward with our analysis. So yeah, we went ahead and we used the gray matter mask mean signal from the gray matter as a reference signal, and we decided the time point plus minus 3 TR that corresponds to 6 seconds. We decided the time points that has the highest cross correlation value with the global gray matter signal, and then we shift the time series to make sure this delay is adjusted for. And this procedure gave us these two delay maps for two groups. You can see that the blood flow starts from visual areas that goes to somatomotor areas. That is in line with the other studies doing the same analysis. However, we didn't find any group level differences between these two maps for brain regions. But when we check the extreme points, you can see most extreme delays, either negative or positive, are statistically stronger in stroke groups. So although on average they show the same delay map, stroke group has significantly larger positive and negative delays, meaning that their maps are more skewed. And for this delay mapping, it makes sense because since each stroke subject has a different heterogeneous lesion, on average, maybe the delay maps are statistically the same, but when we check the extremes, we can see the differences. Then we went ahead and used both corrected and non-corrected time series. And we created the functional connectivity matrices using this atlas that has 400 regions of interest that has also divided by 7 functional networks. So in this atlas, we have we divide the brain into 400 regions, and each region corresponds to 1 functional network. So each subject has now 2 connectivity matrices, one from corrected time series, one from non-corrected time series. To understand the effect of this time delay fixing, we made this calculation on the connectivity matrices named functional connectivity aberrancy. So how aberrant, how deviated and nodes connectivity strength. To be more clear, first we calculated what is the mean correlation value of a node. And then for each subject, we check the standard deviation of the same node. And if it has like if it has like 2 standard deviations away, if it is 2 standard deviations away from the mean or not. And when we calculate this for each subject, each node, on the left side, you can see control subjects has this distribution of 0, meaning that their connectivity values are normally distributed around 0. But stroke patients have this negatively skewed distribution, meaning that they are further away from mean connectivity values. So their connectivity values, correlation values are skewed. But when we check the same thing with the corrected time series, corrected series from the time lag, we can see that their mean is the same, and they are matched. They are matched in terms of the normal distribution. And this tells us fixing the time series for their delay, hemodynamic lag, actually works and aligns them. And for now, we thought it is enough for go on with the other analysis, where we make more control analysis that I'm going to mention later on. And we needed to decide how we want to explore the functional connectivity. And we decided on this dimensionality reduction technique. We wanted to use this low dimensional technique because there are many other metrics that can be used, seed-based connectivities, network-based connectivities, or just network-based statistics. But there will be a lot of parameters to describe, and the reproducibility will be low at the end because of this heterogeneity in design. However, this dimensionality reduction technique is highly reproducible, and it actually summarizes the brain function by 3 components or more. Like it can be, for example, principal component analysis applied on connectivity metrics, or diffusion embedding. We went with diffusion embedding, but the gist of this method is that it gives you the functional distribution of the brain regions in embedding space. And it has been used in many studies. The common finding is that the first component gradient, in this case, that explains the brain function, divides the region's function based on its unimodality or transmodality, meaning that you can categorize brain's function mostly by its ability to process basic functions or not. So in the first gradient, from regions that are red to the other colors, you can see red colors are regions that are associated with higher order functions such as decision-making, introspection, and blue and green colors are visual and auditory areas. So this is how you can define the brain function, like, and it explains around 25-30% of variance in the brain. And in the second gradient from blue to green regions, that explains the second most variance. It is from visual to sensory motor areas, meaning that the brain functions can be divided into two based on their similarity to visual functions or somatomotor functions. This describes the organization in the basic axis, basic modalities. And we did this analysis for our data, and we replicated the results. So the first gradient nicely replicated the unimodal transmodal distinction. The second one explained the visual somatomotor distinction, and the third one explains active passive attention distinction. So when we put all these three gradients all together in the embedding space, we have this distribution of the brain regions in the embedding space, and we wanted to take this as the reference topography, meaning that the brain is traditionally distributed like this for control subjects, and the idea was go ahead and create this for each stroke subject and calculate the difference between each node, each region, Euclidean distance between each region to the stroke subjects and the reference topography. So let's take this region in the corner. The Euclidean distance between this point and subjects, same region of the subject, is giving us a metric for functional deviancy. So the difference between control topography and subject's own region is a description for functional atrophy or deviation. When we calculate this for every subject, we saw that stroke subjects have significantly higher functional deviation to the reference topography, which is expected because of the stroke lesion. And when we check the gradients separately, we saw that the source of this difference is coming mostly from the second gradient, which defines the visual and somatomotor functions, which is the unimodal function gradient, meaning that stroke primarily affects the organization of the basic functions in the brain, not the higher order functions. And when we check the region-wise distribution of these differences, we found functional deviation is mostly functional deviation for stroke subjects are mostly distributed along the somatomotor area and on the right hemisphere. Since the first results were right hemisphere oriented, we wanted to see if this is an effect of the lesion distribution in our sample, and we treated the left and the right lesion subjects differently, separately. And the number of left lesions subjects were 56, and right lesion subjects were 48. the resulting pattern showed that when there is a lesion on the left side, the functional deviation is more symmetric. But when there is a lesion on the right side, the functional deviation is more right hemisphere oriented. So stroke affects the right hemisphere more than it affects the left hemisphere. And we went a bit more in detail of this location of the stroke, and we checked the correlation between lesion of the location of the lesion and functional deviation profile. So for each two subjects, for example, we checked the difference between both Jacquard index and centroid of their lesion, and the correlation between the functional reorganization. And we saw that there is weak relationship between the location of the lesion and the functional reorganization profile. And it is stronger on the right hemisphere only. So when the right hemisphere is damaged, the brain reorganization follows a similar pattern up to some extent. But when you have a lesion in your left hemisphere, the reorganization doesn't follow the same pattern, meaning that a left hemisphere, when it is intact, can manage the rest of the functions in a more systematic way. We also wanted to check the intrahemispheric functional reorganization. And to do this, we calculated the intrahemispheric connectivity matrices, leaving the interhemispheric connections behind, and just wanted to see the organization of one hemisphere like separately. To do that, we calculated the gradients separately for each hemisphere of controls, right and left hemisphere of controls that are both healthy, and right and left hemisphere of stroke patients. And we further divided them into intact hemispheres and damaged hemispheres. So each subject has one intact hemisphere, one damaged hemisphere, and their connectivity matrices are used for calculating the gradients, and their gradients were used to calculate the functional deviation as we did for the whole brain. And we saw that intrahemispheric functional reorganization in stroke patients are still depending on the side of the lesion. When you check the graph here, you can see control subjects have this right-left difference naturally. So right hemisphere has more functional deviation compared to rest in general. So functional distribution of the left hemisphere is less dispersed compared to right hemisphere. And this effect is preserved, but it is higher in the intact hemisphere of the stroke subjects. But this pattern is a bit broken in the damaged hemispheres. And when left hemisphere is damaged, the laterality index right-left balance is broken, indicating that when intact, the right hemisphere cannot conserve the balance by itself. But when the right hemisphere is damaged, left hemisphere helps keeping the same balance as control subjects. Lastly, we checked the functional deviation and their relationship with behavioral impairments. When we received this data set, the authors also shared their huge neurophysiological battery. They had many surveys and questionnaires, and they also had this clustering analysis on the answers, and they managed to cluster the responses into three. And in the first cluster, batteries questionnaires that are related to language, verbal memory, and spatial memory explained the 25% of the answers. And in the second cluster, left body performance visual field bias explained 23%. And in the third cluster, right body visual field bias and attention shifting explained 17%. So all these neurophysiological batteries can be decreased into three factors. And we used the weights of these factors to explain the functional deviation. Basically, we correlated the functional for each region, functional deviation of that region, and the weights of the behavioral clusters. And we saw that big factor 2 that explains left body motor performance and visual field bias are significantly correlated with functional deviation, especially mostly, again, on visual and somatomotor areas. We also had some correlation with big factor 3 and 1, but they did not survive the statistical correction for multiple comparisons. We also checked the anatomical Euclidean distance to location, lesion, and functional deviation profiles. We again found some correlation, but they didn't survive the multiple comparison correction either. And lastly, we made many control analysis for the temporal lag correction to see if they are fit for the whole brain connectivity analysis. And we saw that lag correction does not reduce the differences between groups, which one could expect, because basically by shifting the time series, you are forcing the time series to be similar to each other, right? But for shifting the time series actually enhanced the differences between groups. To me, that can be interpreted as this step actually eliminates the noise and enhances the group level differences in functional profiles. But this is specific to functional related analysis, fMRI related analysis. When we read the analysis related to anatomical distance to location or lesion centroid, we did not see any difference. So this step will be mostly necessary for fMRI related analysis. So conclusions, lag correction increases the alignment between fMRI data of stroke and healthy subjects and enhances the differences between groups. So we highly recommend it. And functional connectivity gradients is a suitable method to map the functional deviation related to stroke. And stroke primarily affects the organization of the basic functions rather than advanced functions. And functional deviation is not, it cannot be only explained by the lesion location, because subjects with lesion around the similar region have different kind of reorganization profile. And this functional deviation can explain the behavioral impairment, and there's a potential to explain or predict the recovery as well. And stroke lesion to the right hemisphere is associated with more asymmetric functional deviation profile and focusing on the right hemisphere, and higher lateralization balance, meaning that if you have your left hemisphere intact, your laterality index will be kept. And future projects, I believe there is a lack of documentation for the effect of stroke on by different modalities. For example, in this study, we explored the effect of hemodynamic lag that wouldn't be obvious from the structural scans only. So I want to go for a software suite that would report the effect of the stroke from different perspectives. For example, lesion detection using T1. This will show us the altered structure of the lesion and will be useful to segment the lesion lesion area. And I think there is a lack of solid toolbox doing this because the available ones are mostly they depend on the supervised models that are trained with the expert drone, hand drone masks. But I think in order to increase the training size and anomaly detection algorithm with guns based on the healthy brains will be superior. And bolt delay detection from functional MRIs, which we applied for this study as well. And they show the areas that have delayed blood flow, but there is not a clear distinction between the blood flow delays due to the regular temporal differences or due to the stroke lesion. So there is this lack of refinement between these two. And another important point is frequency detection. There is another aspect of these functional signals, the anomalies in the functional aspect of stroke, which is frequency. It's not, the fMRI signal is not only affected temporally, but it also affected in frequency domain. There is only one study showing these differences. And I believe this is related to either weak blood flow to the area or showing peripheral support that just causes slow oscillations. And I would like to work on it and also show the frequency anomalies. And if we can fix it somehow, as we did for temporal domain, I think it can further improve the signal. And lastly, since the data set we use also have DTI scans, it's also a good idea to identify the stroke-related microstructural differences. And altogether, these aspects can result in a nice toolbox that can be used by anyone. And they will just need an fMRI scan to get these reports. And the research outputs we created with this project is one journal preference that is under review that we just received the first round of revision. I presented some of the results in Department of Physics in Jagiellon University, and I attended OHBM meeting with a poster mentioning these results. And Wojciech led another work related to effective connectivity using the same data we used here. Last but not least, I also have other stuff going on about blindness, multimodal network similarities, and Alzheimer. I have the references. If you're interested, I can share them to you. And thanks for your attention.
Many thanks, Cemal. Thank you very much. I would like to apologize everybody for our delay. It is, technical problems. So I hope that we still have time for questions, for discussion. I kindly invite everybody present to ask question and suggestion. Well, maybe before it will start, could you please show this slide with your future work? Oh yes. And can you explain what will be necessary to perform this project you propose? I mean, from in the sense of gathering data, maybe it will be necessary to have some equipment, or you suppose that you will have a kind of just commonly accessible data.
For the first one, I will use open data sets that have basically T1 MRI of healthy subjects, but I will need some strong computational power that I think RS will be useful for this. But of course, for the details, I have to spend more time. And if we cannot handle it in RS, maybe we can get in touch with you. But for the rest of them, we already have this data set we used for this project. And the data set has longitudinal fMRI, MRI for T1 and DTI scans. So for now, this should be sufficient. This data set we have should be sufficient. And I also remember seeing 1 or 2 data sets with fMRI of stroke patients online. So apart from the first one, the other ones won't be computationally heavy.
Well, we already have Wisław ready to comment on this question. Wisław, please go on.
Okay, thank you, Marian. Hello, thank you very much for your presentation. I have a few questions. The first question is, have you ever consulted your results and future projects with any stroke neuroradiologist or stroke neurologist?
No.
Okay. Yeah, so your title suggests, because you are dealing with acute stroke, that you are dealing with stroke treatment, because of course acute phase we can treat. If it's chronic, it's another story. But from a clinical perspective, usually a clinician is interested: is it hemorrhage? Is it infarct, and how big? Is it penumbra? How big? And what is the mismatch between penumbra and the infarct? How your work can contribute to clinical approach?
I can work something that will tell you if it is ischemic or hemorrhagic. And this is something that can be also detected by naked eye, so it shouldn't be much of work. And in terms of detection of the penumbra, the detection segmentation algorithm should be able to give the sub-regions like core and penumbra as well. So these two, I think the algorithm should report. I haven't started working on this widely.
Segmentation algorithm to detect it, to get infrared and penumbra from what sort of data? From fMRI board?
No, just using T1. I was planning on...
Sorry, it's completely confusion, because on T1 you cannot see acute stroke. You can see on diffusion, but on T1, if you are using T1, in fact, it's the same comments for your future projects. T1 is not very much useful, and it's not being used clinically. So if you can do it from T1, it might have a sort of publication value, but I don't think so it has any clinical value.
Yeah, I was also checking the accuracies from T1, and they weren't very high. But for I don't know, it is one of the most common modalities, so I wanted to take the challenge actually. but we can also try with T2. I think it's more clear from there, right?
Basically, what is being used is clinically diffusion. It gives you the infarct, and perfusion, which gives you penumbra, and then you have mismatch. Sometimes it's flare, not T2, but flare, because a stroke happened overnight, and you don't know the time from the stroke onset. Then there is a mismatch between diffusion and flare. But T1
I see.
shows you rather a sort of permanent lesion, but I don't think so. It's very much useful in acute phase.
Yeah, we can.
Yeah, go ahead. Yeah.
We can use any kind of modalities, and if also T1 works, as you said, it's a publication. But yeah, apart from the basic research, I should also aim for the clinical usage. So I will keep that in mind. Maybe we can stay in touch, and I can ask like more of your opinion.
Yeah. I'll comment on that soon. Why your functional connectivity is not related to the size of infarct, to the size of penumbra? Why you just consider left and right hemisphere without taking into account the size of these two main regions? And second sub-question, why it's also not correlated to any stroke scales? Because of course, stroke might have different seriousness. So the NIH stroke scale, there is Bartel index, modified ranking scale. Why don't you correlate your work with those scales?
About the infarct lesion, we had this overlap between the subjects, and we just checked the percentage of the overlap between subjects and the similarity of the functional reorganization. But as for individual infarct lesion, since it didn't give me the precise location, I didn't want to use it. But yeah, it's another parameter that we can include in the work. And for the stroke scale, I actually took the NIHSS from the battery from the publishers, and I used them, but it didn't give me a better prediction correlation compared to the big factor of the second behavioral factor. So actually, I tried, but they didn't get a clear pattern, because it is due to the fact that there are 400 regions, and I have to correlate the functional deviation score with the behavioral stroke scale in each of them. So maybe it is suitable for something more general rather than the functional reorganization score of each region.
How much time it takes to process a case?
Not much.
Meaning seconds, minutes?
Yeah, for the plots, yeah, it takes some seconds.
Yeah, because you see, we are supposed to organize a meeting with a radiologist. In fact, he's chief radiologist in Poland, and he's leading radiology department. It's the biggest governmental hospital. And the meeting will be beginning of November. And his interest is, of course, in stroke and in AI in stroke. But as a clinician, of course, he is much interested to have early detection, because if you don't have early detection, you cannot treat, you cannot do much, and it's out of interest. So if you feel that you'd like to try some other data, because I understand that if you're using data from Washington University and you have already something like T1, it's nice. You can do research, you can publish, but I don't think so. It's clinically much of interest. So maybe you can determine what sort of data you'd like to have, what sort of test bed you'd like to have. And during a meeting with him, of course, your project, your work, your future plans can be presented and see if we can align with his interest. What do you think?
Yeah, that will be great. I would like appreciate some guidance. Actually, it's been a while that I'm not getting any supervision, so I will be glad to like hear your opinion and present the work there.
Yeah, that's fantastic. So Kasia will be coordinating. So stay in touch with Kasia and maybe include your your work into that. How much AI do you use in your work? How
In
much?
this one, there is right up until now there is zero AI.
Zero, yeah?
Yeah. It's all linear models. Apart from the diffusion embedding or the gradient approach, but it's not something very.
And do you have any plans to use AI in your work, or do you see
Yeah, for
any?
the lesion segmentation, I was planning to try my chances on it.
Yeah, because this is another avenue. With Maciek, we are going next month to Asia to a big conference. They are interested in collaboration, AI in neuroscience. So if anything, what you have in the pipeline is of your interest, again, try to specify, and it can be included in Maciek presentation and in our potential discussions.
Yeah, that will be great actually.
Okay, yeah.
Thank you so much.
Okay, thank you and good luck.
Thank you.
Well, thank you very much for this discussion also. Well, do we have any other questions? Well, it seems that we have no more questions, but I think that it was a really interesting example of analysis, and well, we'll see to which extent. I hope that the future project which you have mentioned here will be possible to be done. And well, so many thanks for your talk. Many thanks for discussion. And well, we have learned something about what has been achieved and about plans. And well, thank you very much. And I would like to invite everybody to a seminar which will be next week. We will have Filip Katulski, who is a PhD student at the personal health team. Thank you very much and bye.
Bye.